Genetics, dominant and recessive Punnett squares. Genetics is the study of genes, heredity, and variation in living organisms. All genetic information is carried in the DNA. Genes determine traits for each person and species. For example, dimples are a trait just like rolling your tongue can be a trait. Genes come in different varieties or versions called alleles. Each mother and father have their own allele combination. Some alleles are dominant while others are recessive, blend together, or will always show. The purpose of Punnett squares helps us predict the certain outcome of traits in an organism according to the crossing of a mother and father's allele combinations. They show the probability of a trait happening as well as all of the possibilities, and Punnett squares display possible allele combinations from the mother and father allele, and it can help determine the probability of possible types of offspring. Punnett squares will show each trait represented by a pair of alleles. For example, uppercase D is dominant and will stand for having dimples, whereas lowercase d is recessive and meaning you will have no dimples. Individuals have two alleles for each trait. Alleles separate when gametes are made, which is shown simply using a Punnett square, which I'll show you in the next slide. On a Punnett square, the alleles are on each side of the box where they can cross and match up and pretty much are breeding. The alleles separate and pair up within 4 boxes. Each box represents a genotype possible, which shows which trait will be created. A genotype is the actual genetic makeup of an individual for a trait. Here we have a homozygous Punnett square where you can see the uppercase D on top, we'll say that's the father for dimples, lowercase d on the left, which is the mother, meaning she doesn't have dimples. So, homozygous alleles are when each trait is dominant or recessive, meaning they both are 2 uppercase D's or 2 lowercase d's. Now here shows when they cross. You can see that the uppercase D comes down with the lowercase d, so you get big D, little d, same here, big D, little d, big D, little d, same here. So pretty much this means all cross matchings are going to be the same thing, meaning that the uppercase D, the dominant, always overtakes the little d. So in this case, 100% will have dimples. Next we have a heterozygous Punnett square, where the father on top has uppercase D, lowercase d, and the mother has this exact same thing on the left. The alleles are obviously the same, so when you see the possible crossings, they're much different. Here you get double D, here you get uppercase D, lowercase d, uppercase D, lowercase d, and little d, little d. In this case, 75% will have dimples, and there is a 25% chance that they would not have dimples since this is recessive. Punnett squares help to determine different genes and types, and can be used for all different sorts of crossings.